oxidizing or reducing it, and that's the basis. You may get a little bit of that in ecology when you talk about nutrient cycles, but it's still this kind of black box mystery thing. And I think one of the things I want to do during my sabbatical is think about rewriting some of the ideas on energetics. But that's probably more my deal than yours, but if you find yourself really interested in the other trophic avenues, come and talk to me. Maybe we can do an independent study project, and I can learn from you, and hopefully you can learn a thing or two from me. Oftentimes people are shopping for a research project or something for a few extra credits or something. It's a diverse world out there. Well, we know about this idea of enzymes, and we know in general that enzymes are what class of molecules, if you had to get going. Maybe it's up there, I don't know. But are they lipids, are they nucleic acids, are they proteins, are they carbohydrates?
Protein.
That's how we learn it, yeah. Most all of them are proteins. There are some ribozymes that have been discovered that do have enzyme capabilities, but for our case, we're okay with proteins for the most sense. So that's cool. Okay, now, these proteins are kind of special. Of the proteins that we know about, a lot of them are structural and some are functional. Being a functional metabolism, I mean, structure is a function in itself. But and some of this, at first seems kind of magic to us. We go, well, we know that they increase the progress of the reaction or the rate. So this is the rate of a reaction in the red line that would go on without an enzyme. This is theoretical. It's C and D and A and B. So we know that you have to reach an activation energy. We always, we probably learned this. Activation energy, what is that? And then we have that the reaction is progressing over time. How fast is it going? So that infers a rate. So amount of the reaction going on per time. So time and energy are the key factors here. So when you look at any graph, and someone asks you something for the first time, don't get intimidated. Look at what do I know, and these things. Look at your axes. Then look at the relationship, and then start saying, well, what do I know about this? Okay, I know a few things. Okay, I know what a rate is. I know something about energy. Okay, and we're going to move from there. So now we add this enzyme, this magical protein, and we see that the amount of energy needed to make the reaction go is no longer a huge hump. It's a little bit of a hump. You still need some energy to make this thing go. That's the whole key of it. You got to talk about the cell having energy. But the other thing is that we see that overall the rate of reaction, how much product is being produced, is increasing. Also, we've lowered the activation energy, we've increased the rate of reaction over time. And what I want us to do before we move on from this very simple concept, that I put right in there for your notes, and that we see that we have more product in a shorter period of time. Okay, so we know about this. They're highly specific. That's why enzymes tend to regenerate themselves. You don't have to. The cell doesn't have to remake them all the time. But also realize, especially with microbes, that the cell isn't making all enzymes all the time. Okay, there are enzymes that have to be made for things. If we have aerobic respiration, for some of the different compounds in the citric acid cycle. Okay, so they're constitutive enzymes. And then there's also enzymes that are on an as-needed basis. Maybe you get a stimulus. You learn about lactose, the lac operon. Don't have to really go through that pathway if you don't need it. Making a flagella, other things, because why? It's energetics and it's space. There's only so much space in that little bitty 3 micrometer by 1 micrometer cell, okay? And there's only so much energetics to go around. And as you saw, if you're spinning that flagella around, that's taking up a lot of energy that you might need, you might not have more of to make all your enzymes go all the time. On the other hand, if you're not doing enough metabolism, your basal metabolism, as we call it with, I guess, our human health, we're not doing so well. We are suffering what we would call not good health. Okay, reactions, enzyme plus substrate makes it complex, and we get enzyme and product. The key thing here is that the enzyme stays conserved, as we might say in science, that it doesn't get used up. The substrate gets changed to some degree. And then it depends upon substrate binding position, the substrate relative to the catalyst. So if I were to ask you how many different number of structures build a protein, what would you tell me? Would you tell me anything? You say, oh man, I'm drawing a blank. Let me help you. When we just string the amino acids to get the amino acids of the monomers, if we just string those together in a line and we look at the sequence of amino acids, we call that the protein's what structure?
Primary?
Primary structure, yes. So as we move up, above, in general, how many levels of structure are there in proteins? 4, yes. So, if we start taking, and just to review, some of you are going, I'm not sure I'm with you yet. Okay, so if we take the primary structure, and we start making it into a corkscrew or into a beta sheet, kind of like a Venetian blind kind of a thing, we say, oh, that hydrogen bonding is inducing secondary structure. And this should be somewhere in your book if you're saying, oh, I don't remember that. Okay, and then we take the secondary structure, and we look at the hydrophobic regions, and they kind of like being by themselves, and the hydrophilic regions, might be more to the outside of the protein. There's all this hydrogen bonding going on, and then we have cysteine making cysteine, sulfur bridges, and methionine and all that. And we start getting this three-dimensional structure that is above and beyond the secondary structure. That's the tertiary structure. Okay? And then if we get several peptides, if they're tertiary structures together, and they're coming together, they're going to also kind of reform the position of the other or the other peptides, and you'll get a final quaternary or fourth structure. Well, it's these third and fourth structures. You don't always have a fourth structure, but it's the third and fourth structures that really come to dictate the power of the catalyst. I mean, it's important in all protein chemistry, but the catalyst, it's really important. And here we have the catalyst as this, beigeish, brownish blob. Okay, well, we're trying to simplify things. We know in our heads there's a bunch of amino acids in there, all curved around and so forth. And we have here adolase. And adolase has what we call an active site. You probably remember that from days gone by. The active site is where this enzyme interacts with the substrate. Okay? Of all that protein, I mean, they're being concerned. When you really look at the active site, it's just a little bitty area out of all of these tangles of amino acids and stuff, and that's it. All that other stuff comes down to making that site special. Okay. Now, in the case here of fructose 1,6-bisphosphate, we have the sugar structure come in here, and we see hydrolysis or cleaving of that bond. This is what the catalyst does. When it comes down to breaking something, an active site strains the bond. I mean, here it looks like a little wedge comes up and cuts it. Okay, well, this is a cartoon, but in energetics we know that it fits the substrate perfectly, and then it's able, once it's fit, that starts to change the structure of the protein enough where it strains that bond to the point of breaking. Okay, that's it. It's not magic. It comes down to energetics and what we know about basic covalent bonding or whatever the enzyme activity might be. The opposite is true in your forming bonding. To bring the two parts of the compound that are going to come together in proximity where that covalent bond wants to form. It might be in nature, it wants to get there at some point, I'm being very general, or it might not get there at all, there being a bond forming between compounds. Putting them into proximity can change all that. Now, there's no shortage ways to describe this. Those of you in biochemistry have probably learned the Michaelis-Menten kinetics and all kinds of things. And that's why we're going to keep moving, because those courses do such a good job of explaining all that. But we want to remember that no, it's not really magic. Okay, big arrow to help you remember that. Catalysts. Many enzymes contain small non-protein molecules. We talked about this. Those small non-protein molecules, we were calling what in the last lecture? Just to connect some dots here. Well, they can be cofactors, which we might call vitamins, micronutrients, that kind of thing. And therefore, when you look at nitrate reductase, you look at something that it can take an oxidized form of nitrogen and make it into nitrogen gas. And we're very interested in that ecologically in Illinois, because we export more soluble nitrate to the Gulf, at least as well as our other other states in the region. And we're not necessarily proud or apologetic about that, but we are a farming state any way you cut it. But people are very interested in how you can maybe take a wetland or something and then have nitrate reductase come in there and do that. Only recently have I seen papers asking about the availability of molybdenate. And you're like, what? Who would care? Don't you care about nitrate and the microbes? No, molybdenate is one of the non-protein molecules that drives that one enzyme reaction. Now, it turns out molybdenate is around usually enough. You just don't need a lot of it, and the enzymes regenerate it. But now we're talking business. Now microbial ecologists are getting down to, we're thinking this through. It's not necessarily this black box and magic happens. Okay, there's probably magic. I don't know. That's not my field. But here we talk science. Okay, there could be other groups, prosthetic groups that bind to the enzymes. Talk a little bit about that. We see the structure of heme, that really wonderful kind of quadratic structure, and we say, oh yeah, that's pretty neat. And we know that heme groups exist in us as well. Coenzymes loosely bind to the enzymes. Most derivatives of vitamins. Our big coenzyme that we'll be talking about today will be NAD, or in its reduced form, NADH, the shuttling of electrons, and FAD, a flavo coenzyme, flavoprotein coenzyme, just to put away a few more cobwebs. One other thing we should get down pat right away from the beginning is redox reactions. I know chemists, this is a second nature for you, but we should get our terminology straight. We know ATP is the thing that we regenerate. We notice, oh, it looks like a nucleotide. Yes, there you go. Good for you. And we know that the existence of this phosphate, whether it's on or off, is the difference between ADP and ATP. And just shuttling that phosphate off and on turns out to be the energy currency of the cell. It is the three bears. Okay, it is other phosphate groups release and take a lot more energy to form, and some a lot less. This one happens to be energetically favorable, and yet we know we can't store all our energy in our cells or in a microbial cell as ATP. Well, maybe we don't, but to review, it takes up space. It isn't terribly stable when it comes down to storing energy. And those are most of the main factors in ATP. I have a slide that will help us replace that. But when we think about redox reactions, we have an electron donor and an electron acceptor. We'll review glucose in a moment, make sure we have our thoughts screwed on correctly. Electron donor, the substance oxidized in a redox reaction. Electron acceptor, the substance reduced in a reaction. This I find is sometimes hard for students. They either rely on other people to tell them what oxidation or reduction is. I want you to try it. I mean, some things I still have to think, not that I know everything, but I mean, I've been working with stuff for years. You would think I know is what I mean. I still have to think about, iron 2 or iron 3, which one's the more reduced form. And I got to really think about the metals in this. But for most things, we can figure it out. And once you watch where the electrons go, it's not so bad. Here are some of the rules I use, and I can do it really quickly then, and I want to share these with you. Perhaps you have other rules. Oxidation. Well, it is, after all, you're going to be removing electrons, okay? Removal of a hydrogen and addition of oxygen. Reduction is the opposite of that, okay? So we're always looking for that kind of situation. What is being oxidized, what is being reduced. So we have our mythical, not so mythical, our formula. What is that formula? How would you, if I put that on the exam now, which is likely, and I say, name this formula, or this is a summary of what? What might be a really good answer? Aerobic respiration, yes, the key being the oxygen representing aerobic portion. We think air, but it's what we do. So glucose stores chemical energy. It is reduced, okay? It has a lot of hydrogens. Yeah, there's some oxygens there, but when you really break it down, it's really CH2O 6 times. So we see that with the carbon, there's a lot of hydrogens associated, but at least relative to the product, when we see that there's no oxygens on there. So when we see reduced compounds, later on we see NADH versus NAD+. We have to think, well, which one's the reduced form? Which one has the energy that's willing to give away? And so we go that. Oxygen accepts the electrons, it makes water, and CO2 becomes oxidized in this, so that's the where the electrons go. That's where the electrons are lost, and they go into water eventually. And in between, we have a whole bunch of reactions that we'll talk about today. This is just an example. I think we're going to move ahead. Reduction potential. I want to talk about this a little bit. We don't do a lot of energetics and thermodynamics, but I really like going through this, so I will. Reduction potential is the tendency of a compound to donate electrons. Sorry, I even got sleep last night. Measured under standard conditions, it's volts. Okay, it's like 110 volts. It's that. It's not. Oh, those are the volts in the wall, and these are the volts in this. It's volts. Okay, so it's potential. Okay, substance can be either electron donors or acceptors under different circumstances. Really depends upon where the substance is located and what it's giving its electrons to, or its capacity to accept electrons. So it doesn't have to be a magic place. Reduced substances of a redox couple with a more negative E super O prime donates electrons, or a more negative reduction potential, and to the oxidized substance of a redox couple. Okay, so those are words, and they're good to read and go over. I always like to go through examples. Now, in my experience, not a lot of people have thought about the electron tower, and if you have, it's worth looking at again, and if you haven't, it's good to look at it now. First off, you see we have a scale. We have the reduction potential in negative at the top and positive at the bottom. So we have a scale, okay? Like so many things we do in science, we rank stuff. Okay, so this shows the range of possibilities. Now what this is showing on here also is a subset of the many reactions that microbes can do. Many of them, when we look at cytochrome and FAD and NADH and some of the other cytochrome C and A, we say, oh, those are all parts that have something to do with the electron transport chain. I always wondered if that chain was just kind of a random thing, or if there's some kind of common sense that is behind it. So we'll look at that. So the reduction substance in the redox couple at the top of the tower has the greatest tendency to give electrons away. Okay, CO2 can give electrons back to glucose if it had to, and so forth and so on. You can flip those around and move it to the bottom, and it would be the opposite reaction. The oxidized substance in the redox couple at the bottom of the tower has the greatest tendency to accept. Another way of thinking about it is, the electrons are negative, they go to something positive. Is might be a nice way of thinking it. The farther the electron drops from the donor acceptor, the greater the amount of energy. So that we see that if we have the hydrogen couple, the reduced hydrogen, at a minus 0.45-ish or so, going down and finally making water at a plus 0.9, we see that that has a really great potential to move electrons. And there is your answer for aerobic respiration. Okay, why do that when you have nitrate around, or you could develop the system to do that? Because energetically, you can get a lot more of those electrons by moving them all the way down this ladder. Okay, this scale. Whereas here's nitrate. Okay, sure, it's a +5. Okay, nitrate's pretty good to reduce. It's a nice thing for anaerobic respiration, and it's happening, in a wetland or lake near you. Okay, but it's of the two that this is energetically more favorable. So if you have the mechanics, if you're living in the right place and you're adapted to it, that's the way to go. Us eukaryotes caught on to that quite early, eventually, and said, well, that's it for me. The cyanobacteria gave us some oxygen. Let's make the most of that. And now I can make suborganelles and do all kinds of fancy stuff that these other things can't do. Okay, is it better or worse? No, we're adapted differently. That's how that goes. So those are the main things. What else? We have fumarate with this succinate. We see that's a small area. We have cytochrome B over ubiquinone. If you start looking at the different portions of the electron transfer chain, you'll see that they fall in order energetically. Okay, now first of all, okay, you figured that out. It doesn't matter who figured it out first, but you get there's some reason for the order, and the order has to do with the amount of energy left in the electron, and you can still tap out of it. I just make little jumps in the cytochrome or electron transport point, but those jumps have to be in order. Okay, I'd rather impress upon you that than have you memorize all the cytochrome structures and all the heme groups, and we're going to take a look at them. But to think about energetics, the key in science is when you can, you have more information than you can ever process in your life. That's why we're all carrying these things with us. Oh, when was Aristotle alive? We don't memorize that stuff much anymore. But if we can look for pathways, if we can look for pattern, that's what I'm trying to do with you. I can ask you to memorize all kinds of stuff. I think somewhat, I think there's other ways to spend your time, and that's my philosophy on teaching, so let's start doing that. Okay, arrow is showing what I just talked about. Some things are important to us. Again, I want to point out NADH here. We have NADH plus here. Okay, here's the plus, and we see that there's 2 hydrogens on NADH when it's reduced. So with the 2 hydrogens, okay, plus 2 electrons, we have a shuffling of that. We also have another hydrogen associated with that amine group. So it's moving around roughly 3 electrons. That's its thing. It shuttles all day. It's the taxi cab of the citric acid cycle. Okay. What are you doing today? I'm taking some electrons out to the cytochrome system, and I'll be right back and get a few more. And that's its job all day. Now it's a big molecule. It's fairly handsome. It has some nice ribose things in it. But that is the deal. It is an electron carrier, no doubt. So what should you know? Well, you should be able to recognize nicotinamide, but more so remember its job. It's an electron carrier, and NADH is the oxidized form with more hydrogens on the NADH2, wherever it is up there, that is the reduced form. So the citric acid cycle is going to load up what? More NADHs or more NAD pluses? You want to build reduction potential. Now you can either think back to when you learn the citric acid cycle, oh, I know the answer, or you can think about it logically and reason it out. You know, the purpose of the citric acid cycle is to build reduction potential, to get electrons that you can shuttle over to the electron transport chain. So which one's going to do that better, NAD plus or NADH? NAD, yeah, because it's more reduced. Think of reduction as the chemical battery. Okay, glucose is more reduced than CO2. Okay, it has more hydrogen, more battery stuff that, you know, plants can get that light, you know, that energy from the sun and pack in there, or we're going to release it a little bit as we go through energetics, the heterotrophic respiration. But look for that, you know, who has the reduction potential? Okay, nicotine, adenine, dinucleotide, and we talked about that. I'm getting ahead of myself. So essentially, NADH reaction 1, electrons oxidized form, they react with the active site of the enzyme, the electron donor, and then you have the reduced component. We're going to stay at that kind of general level in our discussion today. So, and it's recycled. Okay, energy-rich compounds. I talked about this a little bit. This is nice. Okay, great. I love chemistry. I love drawing and stuff. It's real fun. I'm a picture learner, so I tend to remember pictures better than I remember names. But somehow I make it. But here's the deal to look at. Okay, we have phosphoenolpyruvate with the endergonic result of creating energy, 51, 52 here. And then we get down to glucose 6-phosphate, the beginning of the glycolysis cycle, one of the first steps. And we have 13, and here's AMP at 14. Oh, well, I want to take one phosphate out to make ADP. Taking the other phosphate out doesn't really give me a lot of energy, right? So in here we have the ATP, ADP thing, again, right in the middle. That's the point. Are all bonds, created equal? No, they kind of look the same when you draw little lines and stuff, but artists and scientists say, well, these anhydride bonds, they're more energetic. We're going to make a squiggle. Is there a name for that squiggle in English? Okay, let's go squiggle until we know what that is. So, but they're saying these are more high energy than we get over here, this ester bond and so forth. And so my point here, ATP, it's the 3 berries. It's just right. Now this is great. You get a lot more energy off of this, but it takes more to break it. It takes more to make it. Okay, and so that is a constraint. It becomes a bottleneck. Okay, this, you're going through all the work and you're not really getting much out of it. I mean, that's great when it happens, but it's not really for us. ATP is doing it, but what? It's a little bit unstable to store for long periods of time. One of the ways you can see if the cell's not viable is look at its ATP level. You can measure it. Okay. But the other thing is that it's not the smallest molecule. Okay, we're going to look in a minute when we put on a simple acetyl group onto palmolic acid. We say that can generate nearly 30 ATPs. Yeah, it can, because we're entering the citric acid cycle. So the body says, well, I'm going to make some fatty acids and some lipids and store those ATPs rather than storing 30 ATPs for just one double carbon moiety of palmolic acid with 16. I could get I can save the space 8 times over that I would have those. So what? The space of 240 ATPs. There it is, bigger. long-term storage, just to connect a few ideas, we talked about storage in plants mainly, not so much us, storage lipids, simple fats. Okay, the reason for that, just went over that. In us and other things, glycogen can be present, glycogen, we know is in the human body for sure. The polybeta hydroxybutyrate and other alkanates, we talked about those as what in some cells. Where did we see that before? Again, I'm trying to connect some dots for us so that the exam isn't just a surprise. I haven't thought about that for a long time. What part of when we talked about cell structure, did we see polybeta hydroxybutyrate? PHB, probably called it. Was it in the cell membrane? Was it part of the flagella? Was it a figment of Mike's imagination? Look at the part on inclusion bodies, okay? Go back and look at that, but some cells say, well, you know, I don't have a lot of room for all these ATPs, and I can't really make lipids, okay? Now, algae are great at making lipids. They're great at diatoms and make lipids and oils all the time. They have no problem with that concept, or some do store it, okay? But some bacteria say, well, I'm gonna do that. And then we talked about elemental sulfur. This is kind of a hard thing to think about, because, you know, okay, I kind of get that this can go back, you know, lipids, they go back into the citric, I kind of get that. What's this deal about sulfur? Okay, and it's only because sulfur is the middle of the road, right? Of the middle of the oxidation range of the sulfur compound. You know, with sulfide on one end and sulfate on the other end. So if you have sulfur, you can still oxidize it more. So some things will do storage that can still produce ATP for them. That one's a little bit harder to understand. We'll return to that more when we get to chlorobium and some of those organisms. Those of you guys in lab, continue to look at your Winogradsky columns. You have organisms that are green in the bottom, so they're anaerobic, and actually if you do pull those out, you can find inclusion bodies in their careful microscopy. Green on the top is probably cyanide, but then some of you are starting to see yellow white deposits, and that's other organisms that say, I don't want the sulfur, just get it away from me, and they're depositing it on the other side. Some of the major sulfur deposits in the world were done by prokaryotes. Okay, now let's get into the nitty-gritty of things. Okay, making ATP. That's our first job. That's what we're going to look at today. We're going to review that process. That's it. Okay. We don't care about calories so much, you know, and you know, we're burning a log of glucose, which is what it is. You know, we try to measure things in heat. We're not going to do that. We want to know about ATP because that's what the cell can use. And we have two different mechanisms that's going to be a part of it. And we have two main processes that we're going to cover. The first part, the first process that we want to talk about is fermentation. This is the result of glycolysis, which isn't up here yet, but it will be. And ATP is produced by a process called substrate level phosphorylation. Anyone want to explain that to me? Boy, that's a lot of words. Go ahead. I know you want to tell me. Well, break it apart. What do you know? What do you think phosphorylation means? A little louder, please. Yeah, you're dealing with moving a phosphorus around. Okay, phosphorylation. Okay, you got that. What's a substrate? We just talked about substrate with the enzymes. Was that just a word on the screen, or did something come up in your mind? Oh, a substrate. Yeah, it's a compound. Yeah, compound's eventually going to, in that case, it's going to bind with the enzyme. Yes, very, very good. Okay. So phosphorus is being moved at the level of a carbon compound. In this case, it's a carbon. Okay. That's it. There should not, cause you a lot of upsetness. You should get that. As opposed to in respiration. Okay, we're going to make ATP directly from moving some of the compounds around, but primarily we're going to build reduction potential. We're going to get those electrons and use them to pump protons out of the cell. The proton pump, okay, called the cytochrome system, and that's going to create proton motive force. What's that? It's going to make the protons want to move because they are osmotically out of balance. It's a chemical osmosis. It's not water. Water is a specific case of diffusion. All right. So that's the deal. To do that, we're going to make phosphorus through oxidation. This is the electronic part of respiration. By moving electrons and protons around, we're going to make phosphorus. Does that sound anything like this? No. I will tell you. We already know on the test where you're going to draw the cell membrane. We've got a few other things we've been talking about. You're going to be doing this. I'm going to ask you about this, okay? In some level, be able to explain it to me, okay? You can study for that starting right now, okay? You can have that in your pocket. I have a cartoon here to help us with this, stolen shamelessly from the book. But here we have, right away, look, we have A, B turns into C, C turns into D. Those all represent, in this case, carbon compounds. In a moment, we'll be changing the theoretical A, B, C, and D, and looking at things with much longer names that describe them. But what do we have? We have here the rearrangement of bonds inside of the substrate. That's what's going on. Just little bitty energetic pushes. I'm going to put a double bond here. Okay, I'm going to move this one over here. Now I'm going to take this little bond and click it over here. And now it's energetically favorably to just put this phosphorus on. Okay, so I have something that looks like C. And now it's energetically favorable. Maybe I have to shift a bond or two, and I can get that phosphorus enough energy, 31 or so kilocalories per mole, to get it onto ADP. Okay, boom, bing, 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 bing. Okay, we have an ATP. So it's the rearrangement, the coupling and rearrangement of bonds so that you can not only get phosphorus, but then phosphorylate ADP. I realize I'm really standing on this, and some of you may be impatient, but I do find people get very confused with this. So from experience, I'm going to kind of continue to jump on this, because if you get the pathways that's going on, you get energetics. Now, here's the battery thing. Production of ATP at the expense of proton motive force. At first, proton motive force, what does that mean? That means is building up protons on one side of the cell membrane, okay, and not on the other, so you have an imbalance. What does everything in the world want to do energetically? What are we all moving towards? Randomness, equilibrium. Okay, so even if nothing else is done, you build that up. They would try to find a way in. The cell's smarter. It puts a little gate there and says, well, you can come in, but you got to give me a little bit of energy when you do that. So we have oxidation, the loss of electron from electron donors. Okay, those donors are the coenzymes that are shuttling electrons, reducing NAD plus, FAD plus, and doing that. So you can think, we start with a non-energized membrane, and we start taking the membrane and bouncing electrons down the cytochrome system, the electron transport. They're getting a little energy and spitting out a proton. A little more energy, spitting out a proton. And so you get a really energized membrane. I mean, in a way, it's like a mini version of what your nerves do when you get action potential. Okay, but this is electricity. Now, I want to ask you a microbe question, and a cell question. Where does this process take place in your cells? Where is your Egg McMuffin turning into ATP or whatever you had this morning? vegetable soup, I don't care. Where are you having oxidative phosphorylation going on and whatever cells are doing that? Probably not your fingernails. Mitochondria, whoever said it and probably said it over here, good. Great. Okay, mitochondria. What's the cool thing about mitochondria? Well, what is it? It's double membrane, right? So as you pump something out, those electrons can kind of stick around and go, okay, come on back, and they come on back. And so you have localization, so compartmentalization increases efficiency. Okay, yay for us. Woohoo, I'm sure glad that proteobacteria jumped in there and did that, 3.5 billion years ago. Woohoo. Okay. What happened, where is this going on in E. coli? When it's making ATP, where is this going on? They do not have mitochondria, as we all know. Where is this process going on in microbes? Cell membrane. Now, what's stopping the cell membrane, or those protons, from careening off? I mean, they're nice and positive. They would love to go off into that polar solution around them, the media or your intestines, whatever. What's stopping those protons from just going away and the membrane going, nothing happening? Well, they're going through that. That is the transition. That's the neat thing about the phospholipid bilayer. Not only does it, contain to some degree, it's kind of flimsy, but it's that transition. Protons can move through it with a little help from the cytochrome system. But now the protons, and this isn't showing, this is the double membrane, the lipid bilayer here. These protons are going outside of that. What keeps them in there? Or do they just go away and just has to be enough of them so there's a big proton heap that they all draw from? How does that work? I've had full professors ask me this question once they start thinking about respiration, but you guys know the answer. You're thinking. Well, let's back up a little bit. What's exterior?
Does that have to do with the charge difference between the inside and the outside?
Not so much this way. This makes it look like positive and negative, and it's a big magnet. But this might as well be the English Channel, charge-wise. I mean, because you're going past two really long phospholipids, and there's charge separation. Good idea. Good guess. You're thinking about it. Let's back up. In both of our gram negatives and gram positives, what is external to the cell membrane? You've all learned this now. Peptidoglycan and or the cell wall in general. So when I said that the periplasmic matrix is important not only for enzyme degradation, here's another reason. If you have that other layer through there, you have an area that you can charge up, not lose it, and also bring it back. You have to have that area. Okay, that's a big deal. Okay, whether it's complete peptidoglycan from the gram positive, as we learned, or there's an outer membrane, a little bit of peptidoglycan, that's fine, but you gotta have that localization. So now the cell wall, to us, takes on another function. We know that the lipid membrane is kind of puny, it's really cool, but it can break apart pretty easy. Okay, we know the cell wall protects. We know it helps it to stick. We know it contains it helps enzymes. We know it helps to transport certain molecules and so forth. Great, as does the membrane. But now we also know that energetically it's really important to localize that charge because bacteria may have been pre-mitochondria, especially the proteobacteria, but they were still bacteria before they were mitochondria. So you got to have that. You see the deal here? Okay. A lot of people, when you start thinking about microbes, they miss that. You might be going, oh, okay, so what? It's a pretty big so what. Cell membrane, cell wall becomes a little bit more important to us on that. So, okay, let's start going through the pathway. I'm not going to ask you to memorize structures, as I didn't, probably some of you may have to do for other classes, that's fine. We're gonna look at pathways, we're gonna look at things that we can say about things. The first part of the breakdown of glucose is glycolysis. One thing I want to emphasize here is something that Streptococcus is doing right now in your mouth. It's breaking down glucose, you know, I don't know, maybe from the sugar in your toothpaste. I don't know, but it's doing that right now. If you listen, you can hear them. Okay, you can't, I was just kidding. But the thing is, they're oxygen dependent. They do glycolysis, that's it. People say, well, you know, glycolysis is anaerobic. Glycolysis doesn't care if there's oxygen present or not. It just doesn't. Okay, that's something else to get in here. The first part, glycolysis, where do you see air? It's nowhere. So those guys go, well, I'm just going to stick right with that. I don't care about fermentation products. I'm going to get a little bit of ATP. That's okay with me. I'm just going to do that a whole bunch. All right, works for them. All right. So anyway, yes, it could be anaerobic. It's actually oxygen dependent. I think that a lot of the books call it anaerobic. I say oxygen dependent. That's what it is. Okay, so we have glucose. Okay, we gotta energize the system. So we start glycolysis in an energy debt. All right, so we take out ATP, make it ADP. Now we have glucose 6-phosphate. We rearrange the bonds, and actually eating up another ATP, we take that phosphorus and we put 2 phosphoruses on fructose 1,6-diphosphate, and there now we rearrange bonds from glucose to fructose, and we put on more phosphorus. That's the big deal. So what are the key things here? Rearrangement of bonds, phosphorylating the initial glucose compound, although it's something different here, and it takes energy. Those are the take-home messages of that. Will I ever ask you, oh, draw me fructose 6-phosphate? No, I'm not going to do that. In fact, if I ask you anything about glycolysis, people are always worried what's going on in the test. I'll provide a diagram for you. I want the Texaco roadmap of the thing. I don't need to know every filling station on the way. Okay, I just want to know how to get to St. Louis. All right, so now we're in these are stages. So that's kind of the preparatory phases, getting the phosphorus on the rearranging bonds. That's part 1. Part 2, making ATP and pyruvate. Pyruvate's a key molecule. Pyruvate is something we should remember. So now at this point we have a split of the 6-carbon molecule into glyceraldehyde, 3-phosphate. So 2 3-carbon molecules, and then we're able then to get electrons and then put them onto NAD, thus reducing NADH and producing 1,3-phosphate glycerin. Okay, so what has happened here? We basically have been able to reduce NAD plus in the second part, and in a moment we'll make pyruvate. All right, and we've broken up the big carbon molecule. That's what's happened. Now, that's fine. What I want to draw your attention to right now, because we're in microbiology class, is this right here. This NADH, if we go into aerobic respiration, we go into the citric acid cycle, we can use this. This will come along and give us reduction potential. If we go into fermentation, okay, the anaerobic process of breaking down pyruvate, this will actually be regenerated NADH, and it will not produce any energy for us. Okay, that's the big deal of moving pyruvate into ethanol or something, is to regenerate NADH. Okay, so keep your eye on that ball there also. So here at this point, finally we're able to get a phosphorus off of one end of this molecule, because this is adding things up off of a glucose molecule. So there's 2 3-carbon molecules, so we can get 2 of those, we get 2 ATPs. This is where we start winning in the whole thing. And we move through some other bond rearrangements and finally make it. And phosphorus there, we want to get rid of that because pyruvate has no phosphorus, and we get 2 more ATPs. So, let's think about this. We started out, we had to prime the pump with 2 ATPs. In the last one, we got two ATPs back, so we're at 0 at this point, okay? Well, we have NADH, but if we go into fermentation, that doesn't help us. And so here we finally get a net output of 2 ATP. And you say, why does yeast bother to make alcohol except to make the masses happy drinking wine on Saturday night? And the answer is, it's the only game in town, okay? When it's got sugar and it goes anaerobic, it works. It gives it enough ATP to continue to ferment. Fermentation is not the fastest way to break down glucose. My Bic lighter and a piece of paper is the fastest way to break down glucose, but this game works. So once we get pyruvate, there's several different ways we can go. I, again, am not asking you to memorize anything except know that there's 3 different pathways. One, lactate. Okay, we can get the production of lactate from pyruvate, acetaldehyde, which goes into ethanol. Actually, these 2 are enough. We'll pick this one up when we look at the assumptions in the cow rumen. But for this test, know that we can get lactate or ethanol. What's the difference? Can everything do all of it? No, different microbes are doing different things. Okay, you and lactic acid have a relationship. A lot of your athletes probably felt blood burn. Okay, you're breaking down sugars anaerobically, and you're getting lactic acid formation. Okay, that's glycolysis. So pyruvate is kind of our, so my little guy there says, there you go. There you go. So we won. We got 2 ATPs. We didn't win big, but we won. Okay. Here now is the big deal. If you take pyruvate to the citric acid cycle, things change. And it doesn't do both. Okay. Some people say, oh, I do fermentation, then I go into aerobic respiration. No. It depends upon the pathway, the mechanisms you have, and the conditions you're living in. E. coli can ferment. Okay, you take away oxygen, it goes, oh, I'm going to ferment now. Some things just go dormant. You give it E. coli air, and we'll see this in lab today, I think rather nicely. It'll grow like crazy. In the same amount of time, if I put, and we did this in lab, if I take glucose and E. coli, I put them in an anaerobic chamber, they will grow, but they'll grow like 1/10 the amount that grows aerobically. And you're like, oh, they just like air. Well, you can see that, but as a scientist, you go, well, they're producing a hell of a lot more energy, they can make a lot more stuff, and we get that there's a reason behind it. Can it do both? It's not doing both. Okay, it's shifting. E. coli can also reduce nitrate. It's a fantastic little organism. So, I'm saying if we go to citric acid cycle, we're not entering fermentation. Okay, and no, I don't have too many dollars to do. So this is our culmination. This is a slide that adds up the ATPs. The next few slides are just summarizing this. Okay, this just shows the whole process. So stage 1, rearrangement and taking in energy. Stage 2, more rearrangement, 3 carbon moieties, getting some ATP and NADH, which recycles in fermentation or comes over to stage 3 if we stay in fermentation, and then production of pyruvate, the last stage being getting some ATPs. Fermentation, yes, or aerobic respiration. This you will probably hate. When we used to teach with overheads, I did this. The reason I keep this in here is some people really like to look at what happens where, and I also like the way that it shows how you break up the number of carbons. And some people learn that way. They like that kind of didactic reductionism, and that's okay. There's nothing on here that isn't in the last one. Okay, it's just for people who like to know a little bit more and teach it this way also. This is in the summary thing, just showing that we're winning with the amount of ATPs. We talked about that, summary for your notes. And that's pretty much what this says. This also adds the two different pathways I want you to think about. You could add other organisms here. Those are not the only organisms. Stretching, good idea. Keep oxygen going to the brain. Okay, aerobic respiration, oxidation due to oxygen. Okay, aerobic respiration. Okay, higher ATP yield, ATP produced at the expense of proton motor force. We've been through all that. It's reviewing. Sometimes seeing it more than once helps. We need to have a membrane. Now we got that one in our pocket. Okay, transfer electrons to an acceptor, conserve the energy. Some energy is conserved as the electron moves. This is the magic of it, that the electron tower works, just bleeding off a little bit of energy each time, but enough in the end to make ATP. It is, I think, amazing. And then we're looking at the portions of these. Okay. There's so many versions of this. I can't remember where I got some of these. This is an earlier version of Brock. Okay. Well, let's take a look at a few things. This shows the electron transport system going from top to bottom. Okay. If again, I think there's other things to do than for you to reproduce this on paper, although I'm confident all of you could. I have no doubt about it. But when we look at, we have NADH coming in at the top, we have an enzyme that's helping then deplete off, reduce NAD, move electrons in there. You see it's two hydrogens, okay? Those are full hydrogens, those aren't protons. So it's deleting the electrons off the hydrogens. Go back to the NAD cartoon and show you that they came off of that top nicotinamide molecule. So here we have two electrons moving down. We have a flavoprotein that's getting enough energy to move two protons to the other side. Okay, that's kind of cool. Now we get into a heme group, a heme iron group. Okay, some stuff going on over here. Water's breaking up. Go into a quinine group, pumping out two more electrons. This is the outside of the cell. Here's cytochrome B. Here's cytochrome B and C. Here's cytochrome A and cytochrome C complex, and we get hydrogens pushed off there. Okay. The key thing at the end is oxygen. Oxygen, we call that the terminal electron acceptor. And it makes sense now. It's the termination where the electrons end up at the end of the electron transport chain. If you're doing nitrogen reduction, nitrate would be your terminal electron acceptor. This is just going through what each one is doing. NADH accepts two electrons, two protons. It's able to make 3 ATPs when we do our calculations in a little bit. Flavoproteins, 2 electrons, 2 protons. They're not quite as energetically favorable. It's just the way that they're laid out. Cytochromes, they're neat. They have the heme group. You've probably seen this in physiology or other classes. I just show it because some people say, well, why do I need iron? You know, or why do bacteria need iron? They don't have blood. No, they have heme groups. So it kind of connects, some of those nutrient dots for us. And the structure is really neat, but I'm not going to ask you to draw that. We have iron sulfur proteins, very simple, but these complexes are very structurally stable. Quinones accepts electrons, protons, but only passes on the electrons. So quinones are an intermediate. Present. But here's the bang, bang, bang. I'll wake up for a minute. This is an important one. Okay. Once again, we have the electron transfer system, flavoprotein, okay, NAD coming in, okay, iron sulfur group, cytochrome complex. Okay, so the order is still there. Maybe you already memorized that in biology 1. I don't know. Inside and outside. Okay, protons moving out. But look at the electron potential in these reactions, okay? it's still following those rules of the electron ladder, right? The electron ladder we talked about 7, 10, 15 million slides ago. We're going from negative to positive, okay? If you look at that reaction, you go back and look at it, you'll see, that the potential that you can calculate thermodynamically holds up. But more so, when you get to this point, you find this positive potential, and once to find a place for the electron to go to, there's a nice negative component to the oxygen, and it allows itself to be reduced to water. Okay, so that's why there's this order to it. Okay, because of the way the electron has potential to move from one carrier to the next. Okay. You can shoot it all the way down. You can make the reaction go. We can make water. Under the right circumstances, you can make water. But with hydrogen, the guy, the Martian, everyone see that movie? He's making, water and oxygen blowing up his tent and all that stuff. You can make water, but the thing is this little bit of release, pumps all those protons. All right. This is just another cartoon. I'm not going to really explain. Some people like things a little bit more general to see where the coenzymes are, where the cytochromes are, and so forth. We're going to focus now on this next portion of it. Okay. After we have these protons on the outside, after we've made water eventually from what's left of the electrons, this is a hydrogen atom, a hydrogen, two electrons in the hydrogen. We have all these protons on the other side, so what have we done? Some people will call this proton moment force the process of chemoosmosis. Okay. Osmosis you get, it's like diffusion on water from high concentration, low concentration. This is the osmosis. It really should be called ion osmosis, but it's called chemoosmosis. I guess it's easier to say. Anyway, so these guys want to go back in there. Okay, that's the deal. That's what you've done. You charge this membrane a little bit at a time. Now it's time to pay the piper. And we're going to do what the cell says, okay, yeah, you come back. But I want you to go through this channel, and when you make it to the other side, you give me enough protons, roughly 3 or 4, to put phosphorus onto ADP. And that's the payoff. And that's what that looks like. Yes, cartoons are nice. This is the outside of the cell, proton into the channel. Several different proton components to this, different chains, and you're able to make ATP. There's all kinds of summary sheets, should you know. Citric acid cycle pathway through which pyruvate is completely oxidized to CO2. That's what we're going to talk about next and put this thing together. So up there's pyruvate. Where do we get pyruvate from? And the class resoundingly said we got it from the process of glycolysis. And how many pyruvates come off of one glucose molecule? 2. Okay, so we have pyruvate coming in 3 carbons. That's kind of cool. We have NAD with coenzyme A, and we get a reduced NADH right from the beginning. Now we have acetyl-CoA. Okay, it's still two carbons. A very important compound. A compound that is the basis of many dissertation defense questions because it's pivotal to life. If you had to feed one thing to almost anything, pyruvate or acetyl-CoA would probably work, wouldn't it? If you think about it, it's really an amazing compound. Okay, we lose the coenzyme A, and we put a 2-carbon compound onto a 4-carbon. Can you see that? 4-carbon is the color code. And now we have what? How many carbons in citrate? 6, yeah. Triacetate. Okay, so we have citrate. We have some movements of molecules. We knock off a CO2 as we go into ketoglutarate, and here's where CO2 comes off. We need to see how we get carbon dioxide generated. And so we get our first NAD equivalent. I don't care if it's NADP or not in this class. Okay, another NADH here, another CO2. Now when we go from succinyl-CoA to succinate, we get a GTP, a guanine triphosphate. That's equivalent to an ATP energetically. Okay. So, looking at things, we could talk about that. I can get my arrows caught up. NAD, more NAD up there. Okay, GTP and FAD. So we get another carrier one that's not as effective as NADH, but it does a good job. And CO2. Okay, now, our compound, our summary formula says that you get 1 glucose and 6 oxygen molecules, and I get 6 CO2 and 6 water. I'm only getting 3 CO2. What gives? 2 pyruvates. Good. Yep, so we do it again. You know, that, and always look at the question. I mean, whether you're taking the MCAT, whatever people do, make sure you're saying from glucose or from pyruvate or whatever, but that's how people catch you on it if they're doing the math thing. So if we're converting, here's the deal. You give me $1, I give you 4 quarters. That's the equivalence, right? That's what we do is change 1 GTP, 1 GTP, 1 ATP, 1 FADH, makes 2 ATPs. Remember, it's not just a simple conversion. There's protons and electrons moving around. It's all got to add up energetically, and this is even a rough estimate. Okay, probably in some of your cell classes you've learned that. But if we add it all up, for aerobic respiration, glycolysis gives us more than 2 ATPs because we have 2 NADHs that are formed. Go back and look at it. So 2 times 3 ATPs per NADH, I got 6. I get 8 ATPs. Okay, and if I add the GTPs, okay, I get one of them by 1 turn of the pyruvate. Okay, and then I look at the 4 NADHs, the 1 FADH. Okay, that's for 3 carbons. I do it. That's 15 ATPs. I do it again for the other pyruvate. I get 30. I can get 38. Okay. I'm curious, in your other classes, when they tell you about aerobic respiration, what did they tell you that the ATP net production is? 36 to 38. Most people, did they tell you why the variation? Okay, it's okay. This isn't like, oh, you're bad, I'm good. I'm just curious. The deal is, and this bothered me for a long time. Something bothered me, I check it out. It turns out, the story that I have, you can ask your cell biology or biochemistry people, see what they say. In bacteria, they get to 38. Okay, they got it. The 36, as I understand it, is the transport, the energy transport of moving things across the membranes in the mitochondria. That's what I got. That just to do that, there's just a shuttle that it takes energy, eat up, 60 some kilocalories per mole. Okay, I don't know, but see what you can find on that if you should be so curious. That always, it's 1 glucose molecule, how can it give you 2 different answers? And that's what I found from it. For you, it might not be revelation. I thought it was cool. I'm going to keep talking about this. Is it okay if we talk about, we only got 3 minutes left. Is it okay if we talk about the report on Thursday? Okay, I'll try to make the report that we did last year. Don't start working on it. Look at what we did last year. If you go, oh, I want to do that, that's fine. And I'll put some other ideas I'm thinking about. So if you get a chance, look at that, and we'll talk about that on Thursday. It'll probably be a very use for our time if everyone's ready to go. Now, if we get into catabolic diversity, we got this one covered. Okay, we got this one covered. Okay, what is anaerobic respiration? Yeah, it's respiration. What makes it respiration? We talked about this last week now, so think back. It wasn't that long ago.
It has the proton motive force.
There you go. Yeah, you're doing oxidative phosphorylation, I don't care how you call it, chemoosmosis, and that stuff. That's it, okay. We're gonna also talk a little bit about chemolithotrophy. Here, chemical, easy, litho, rock. Okay, this is the neat thing with microbes. And phototrophy. The phototrophy you've learned about is what? Photoautotrophy. What else do you guys call it that you learned since you were, you made your first leaf collection? Photosynthesis, right. There's photoheterotrophy. Whoa, I can do photosynthesis without CO2? Yeah, some things can do that. That's pretty neat. We've talked about this. Here's where I want to end up is on the, here's chemoorganotrophy. You're part of this. Okay, this is terminology. Okay, it's kind of categories of terminology, so don't get hung up. Okay, but we start with organic compound. If we have fermentation, we get a little bit of ATP. We have a regeneration of the NADH. Okay, if we have an organic compound and we have a flow through respiration using oxygen to generate CO2, that's aerobic respiration. Okay, fantastically on this earth, other things can be used as terminal electron acceptors, even organic ones, which in a way is what fermentation is with the NADH. It is an organic electron acceptor. Okay, with catabolic diversity for chemolithotrophy, here this is a little different. You have your carbon being picked up by biosynthesis, usually through autotrophy. Okay, but here you have your inorganic compounds that are still going onto electronegative compounds. They reduce these as they do them, but you have a separate chemical or production of carbon. The last slide on this is phototrophy. We'll start here next time, so don't get carried away on that. But both of these have common to them the movement of energy into the cell by light. This is just amazing that light, coming through the air can get captured into a chemical bond and saved. Okay, we're used to this one. If you add CO2 into it and biosynthesize using this as energy, we make what? Good, glucose. Okay. Photoheterotrophy, this is the difference, that we can still reduce carbon. We can still biosynthesize. You just don't have as much reduction potential to put on that CO2. It's as oxidized as carbon gets, to my knowledge. Right? So under normal circumstances. So organic compounds can serve in that. We're going to finish up this chapter, catch up on a little bit of genomics and stuff. Ourselves